funded
by
the private
industry.
sector, right?
They would theoretically, I am not familiar in any way, shape, or form of the actual legal aspect of this agreement, but I would think that if you're sponsored by industry, that industry would have the first choice or the exclusive rights to the outcome of that research. Does that make sense? So then if we focus exclusively on the private sector, on drug companies, or we're not funding the research, then how is that, we could have limitations in our ability to really extrapolate that research for the general public. I think it's a great thing that the pharmaceutical industry is finally involved. It has been one of our biggest complaints as stem cell scientists to the pharmaceutical industry, and asking them, it has taken them many years to step out and acknowledge and support, and it even goes beyond, in that Pfizer, the company that I used to work for, now has a division of regenerative medicine. Never in my lifetime did I think that the company would move. It's not moving, it's parallel, but they are a company exclusively of small molecules. They create drugs, pills. But now they recognize the value so much that they have a division of regenerative medicine. And actually the head of that division, who's my former boss, is arriving in Madison today, and he's going to be here. He's going to be speaking at the World Stem Cell Summit. John McNeish.
This is kind of a takeoff on the same question. You basically work for the university, and you have this corporation.
Right.
I mean,
we have a conflict. So me, myself, and many others at UW. So we have a conflict of interest committee, and any given time, it's open to the general public, where we declare conflicts of interest, if any. So we declare associations. In this case, I'm a co-founder, so I'm a shareholder of this company. So it's declared in the conflict of interest, and then there's a committee that oversees if there's any conflict between the research that I do in my laboratory with the company. And then they issue, if there is, they issue conflict management programs. So this has actually been, I would say, one of the fortes of this university, one of the most positive aspects of this university compared to other schools, is that we are able to translate discoveries into products. We can't do it at the university. Having worked at a drug industry, we do not have resources at a university to create and deliver new drugs to patients. We cannot execute clinical trials. And so the creation of biotech companies is one of such mechanisms. And Jamie is also associated. Jamie's a co-founder of, Jamie Thompson, of 2 other companies. Tim Camp is also, it's all very open. So I think, I don't know exactly the link, but it's all open to the public, the conflict of interest. Outside activities, outside activities. Go ahead.
You did state that a person's own fat cells would be beneficial in usage for MS?
Yes. Well, yes and no. There are stem cells in fat cells. There's a rich population of CD34, that's the marker, cells that are positive for CD34 in fat cells. I'm not specifically familiar with studies from those cells into multiple sclerosis. But again, so I've seen studies where we've generated bone and fat are very close lineages. So you've generated bone cells from stem cells from fat to replace or potentially as a therapy, for example, for osteoporosis and bone loss. And then there's a company in Michigan called Astrom that actually has a trial for bone repair. It could be bone marrow-derived stem cells or fat stem cells, but there's a lot being done in fat stem cells. You see the limitation again is that you can't expand these cells indefinitely, even the ones you excise. So potentially, for your own use, potentially, research would have to show that you could extract fat from your own tissue, differentiate. Again, these cells are multipotent, they're not pluripotent, so they don't differentiate into all the cell types of the body, and then create cells for transplantation back into yourself.
So what would be the best one for them if it's not fat? Is it bone marrow?
We have beautiful work being done with human embryonic stem cells and bone marrow stem cells as well. So actually, Ian Duncan here at the UW, he has a research program on multiple sclerosis. Ian Duncan, yeah, he's at the Red School. I'll take your last question just because I don't want to hold the other, and then I'll be here and available for other questions. Go ahead.
I'm just curious how specific your neurons get. In your research, do they get down to like basal ganglia cells, or are they just primitive?
No, we've shown, I'm very interested in the 5-HT, the serotonergic neurons. So we have shown that, at least in terms of neurotransmitters, I haven't done markers for basal ganglia. I've done markers for neurotransmitters to say that in those populations, we have GABAergic neurons, we have serotonergic neurons, we have glutamate secreting neurons, and then we have the basic marker of a mature brain cell, which is beta-3 tubulin. We show that, and then we show a more specific that we have subpopulations of different brain cells by their neurotransmitter activity. So that's where we are. Okay, so thank you very much. Thank you for coming.